It's a pleasure for me to be here and teach this lecture on epigenetics at this important lecture series in honor of Casey Brown, who was one of the most influential geneticists and scientists in my career. And so today we will talk about epigenetics. And specifically in this lecture, I've set three main learning objectives. One is we're trying to understand what epigenetics is. Then we're going to try and learn what are the main, the most common epigenetic mechanisms in the eukaryotic genomes. And finally, we will delve into technologies and specifically, next generation sequencing approaches to study epigenetic mechanisms as well. So to understand what epigenetic is, we need to do a step back and understand what gene expression regulation means, because the two things are very tightly linked. And so in the human genome, but in general in eukaryotic organisms that are made of many cell types, you can see that these cell types have different shapes, morphologies, obviously different functions, but they all share something in common, which is they have exactly the same set of genome, right? If you take a blood cell from your organism, or you take a neuron, and you sequence them, they have exactly the same DNA sequences. So obviously, how can you get so much different cell types coming from the same exact genome, the same exact set of 25,000 genes? And that is obviously because not all of the 25,000 genes of the human genome are expressed in the same time in all cell types, right? So you're going to have a set of genes that is only expressed in neurons, and a set of genes that is only expressed in blood cells, and so on and so forth. So you have something called cell type specific gene expression. And epigenetics comes into place here because epigenetics is something that really is crucial to determine which genes have to be turned on and off in a cell type. And so epigenetics literally means above genetic or on top of genetics. And what this really means is that it refers to external changes and modifications of DNA that do not affect the sequence of the DNA, but that ultimately result in modification in the expression of the genes, right? So the DNA sequence is not changed, but instead the epigenetic mechanism, the epigenetic cells, will change the way in which you read these genes and you express or turn on and turn off the genes. So these are changes to the physical and chemical structure of the DNA and not to the actual sequence of the DNA. So it doesn't change which nucleotides are part, will compose your DNA, but will change how these nucleotides are activated or repressed. These epigenetic modifications can be inherited, have to be inherited. And to cite an old paper, but it's still very, very up to date, is that an epigenetic mechanism should be heritable, self-perpetuating, and reversible. And we will see, what this means. And so today, in the lecture of today, we will discuss three main epigenetic mechanisms. One is histone modifications. We will see what histones are and what modifications of the histones represent. DNA methylation, and then we will see the role of long and short non-coding RNAs in the modification of gene expression. So these are the three main classes of epigenetic mechanisms that you can study and that you have in eukaryotic cells. And so going back to the initial conversation we were having, right? So gene expression is a very complex process. Again, we have 25,000 genes in the human genome, but not all of them are active at the same time. Some of them are on, some of them are off. And also the ones that are on could be more highly expressed in a cell type and less highly expressed in another cell type. So how is this regulated? It's very complicated, and it involves a lot of actors. So this one's your gene, right, which is made of coding, portions of coding DNA, so the exons, that will then be ultimately transcribed and translated into proteins. But to determine which genes is active and not, we have a series of components and actors of the genome that will be involved. First, you have some non-coding DNA elements or cis-regulatory elements that can be proximal to the gene, and in this case, they're called promoter, or distal from the gene. and in this case, we're called enhancers. To activate a gene, promoter and enhancers need to be activated themselves, and this activation is normally mediated by specific proteins we will discuss later in the lecture called transcription factors, and by other co-activator protein complexes, which we will also discuss later on. These are all proteins that will facilitate, one, the activation of these non-coding elements. So imagine like enhancers and promoters are switches to turn the light on, right? The gene is the light, so you need to turn it on. And the promoter and enhancer are the switches which you'll have to press to turn the light on. And who is going to press the switches? So who's the finger who's going to press the switch? The finger is the transcription factor. Transcription factor is a DNA binding protein. We will discuss them as well. They will bind your enhancers and promoters, activate them, and then mediate this 3D looping, which will put enhancers and promoters in close proximity. And this ultimately will be necessary to recruit the entire transcriptional machinery, such as the RNA polymerase II, which is the main enzyme involved in transcription, which will then activate the gene and produce the RNA, and the RNA then will be translated into protein, and the gene expression will be finalized. And so, how does epigenetic enter into this conversation? So let's start by saying that let's think about the human genome, for example. The human genome is not big; it's huge. It's made of 3 billion nucleotides, which means that if you take, 3 billion nucleotides, which makes basically 2 meters of DNA per cell, and you have billions of cells in a human body, right? So this is just to say that if you take all of the DNA that you have in all of the cells of your organism, and you tie it up one with each other, you can basically make the orbit of the Earth around the sun 65 times. And this is how much DNA we have in our organism, which is insane, especially if you think how small a cell is. And so how do we fit all this DNA into a tiny cell? Obviously, it needs to be condensed. It needs to be tightly packed. And the way this compensation is regulated is that you have some proteins that are, they're spheric proteins with a specifically structural function. These proteins are called histones, and 8 histones together that you can see here, and specifically 2 copies of the histone H2A, 2B, H3, and H4. So 8 histones in total forming the so-called histone octamer, okay? They would gather together like this, and then you're going to have 146, a very specific number of 146 base pairs of DNA, so 146 nucleotides wrapped around the histone. Then you're going to have a little bit of linker DNA, and then another one of this. So this combination of the histone octamer with the DNA wrapped around it takes the name of nucleosome. So you have your nucleosomes, and when your nucleosomes are tightly packed next to each other, your DNA, your chromatin will be condensed. So what is chromatin? Chromatin is the ensemble of histones and DNA. And I want to repeat myself, 8 histones together, the octamer, with 146 nucleotides of DNA form the nucleosome. And all the nucleosomes together form the chromatin. And when the nucleosomes are tightly packed next to each other, so something I wanted to say, there is an additional histone here, which is called histone H1, which has the role of basically keeping your nucleosomes stable, like this, with a structural function. So then you have all of your nucleosomes here, which is again 8 histones and under 46 base pairs of DNA. And then the nucleosomes, and specifically the histones, can be covalently modified in the histone tails. So you're going to have histone modifications. These are chemical modifications of your histone tails. And like I said before, these histone modifications are one of the main epigenetic mechanisms. And we will see what this means. Basically, depending on the type of chemical modification, covalent modification that you have on your histone tail, this will flag your DNA in a different way. And we will see later on how some histone modifications are typical of repressed DNA, and some histone modifications are typical of active DNA. And to understand a little better some examples of this, we go back into talking about chromatin. Like I said before, chromatin is this ensemble of nucleosomes, right? And each nucleosome is 8 histones and under 46 base pairs of DNA. And so when your nucleosomes are spaced distant from each other with some regions of DNA that are naked like this, your transcription factor can then bind, right, and activate, let's say your enhancer is contained in this region or your promoter. If the DNA is nucleosome-free, if your DNA is naked, we say in our terminology, then your transcription factors can bind and activate the enhancer. And in this case, we refer to this type of chromatin as euchromatin. So euchromatin is when you have relaxed nucleosome composition, some histones are evicted, your DNA is free to move. It's free to be bound by transcription factors and other cofactors. This is euchromatin, whereas when your nucleosomes are tightly packed and condensed like this, you refer to this as heterochromatin, which is a repressive state. So your transcription factors, most of them at least, will not be able to bind these regions. Your other cofactors, RNA-pol2, other protein complexes will not be able to bind. And so your chromatin is going to be heterochromatic or repressed. And this is not just determined by the proximity of the nucleosome, the condensation level, but also by, like we said, the histone modifications. You can see the histone tails here. These are the histone tails of the histones. This. And you can see that depending on the type of flag you have, so these green ones are histone modification typical of active transcription. So you could have H3K4 monomethylation, H3K4 trimethylation, H3K27 acetylation, and so on and so forth. Whereas when you have a tightly compact heterochromatin, you're going to have histone modifications typical of repressed chromatin. And we will see a couple of examples now. And again, this histone modification is really to flag, like a flag that says, okay, this chromatin is repressed, transcription factors have to stay far away, don't bind it, don't activate the genes that are in there. Whereas in this case, you say, okay, the chromatin is active, it's flagged by the histone modification as well, and transcription factors are allowed basically to come and bind and activate this region and express the genes that are found there. And so, which types of histone modification do we find in heterochromatin? To understand this, we need to understand there are two main types of heterochromatin. One is called constitutive, which is heterochromatin that is basically always present in a developed cell. Some regions of the chromatin are always repressed, no matter what. These genes basically are never activated, unless it's some exceptional situation. That's why we say it's constitutive, because this heterochromatin is always, pretty much always in a heterochromatic state, repressed state. And histone modification normally flagged in this is histone H3, lysine 9, trimethylation or dimethylation. So this is a type of histone modification that flags constitutive heterochromatin. So again, these are regions of the genome that are permanently repressed. On the other hand, you can have regions of the genome that need to switch from a repressed to an active state quite frequently. And so you have the so-called facultative heterochromatin. And so these are flagged by histone H3, lysine 27, methylation, dimethylation or trimethylation. The most common being H3K27 trimethylation. Again, this is facultative. So these are heterochromatic regions that maybe during development you need to switch from active to inactive and so on and so forth. So this is the type of heterochromatin that you would have. And something important to remember is that this specific H3K27 methylation is deposited, is regulated by a very important protein complex called PRC2, which is made of multiple different subunits, 3 of which are compulsory, always there. EZH2, which is the actual enzyme of the complex, then EAT and SOS12 are also mandatory. And then you can have multiple subunits that are cell type specific, so you may or may not have them. So this protein complex, PRC2, or polycom repressive complex 2, is heavily involved in depositing this type of facultative heterochromatic histone modifications. And so these are just a couple of examples of histone modifications, but really in the last 15 years, now histone modification has been studied extensively. This was one of the first papers in Nature, 2011, which really demonstrated how during development specifically, enhancers have, in humans, in the human genome, then we found out that it's, conserved across eukaryotes, have very specific signatures as well. You can see some examples here again of histone modification typical of active enhancers. This one is the most, widely studied, which is histone H3 lysine 27 acetylation, typical of active enhancers. H3K4 monomethylation is typical of active and also in poised enhancers. So these enhancers are still not in an active state, but need to be activated quite quickly. Whereas these active enhancers obviously don't show this typical trimethylation of K27, which I just told you is typical of heterochromatin. On the other hand, you have the promoters, such as this one, which present this typical H3 lysine 4 trimethylation, which is instead of a typical histomodification of the promoters. But these are just some examples. There are a very large number of histomodifications that together form what we refer to as the histone code, which is basically literally a language of histone-modifications that is used by the genome to flag specific chromatin states. So you have histone-modifications for euchromatin, heterochromatin, active enhancers, active promoters, gene bodies that are transcribed, gene bodies that are silenced, and so on and so forth. It's really a code, and it's very interesting. It's a very great tool to study, to manipulate the genome. And this is just another slide, just to summarize as well, that, for example, histone H3 lysine 4, depending on whether it's monomethylated, dimethylated, or trimethylated, can indicate different things. Again, poised enhancers, active promoters, or a little bit of both. So it's really a language. It's a code. And it's one of the main epigenetic mechanisms is modifying histone tails. So again, these are covalent modifications that happen at the tails of the histones. Histones being, again, just to summarize, these important proteins, structural proteins that form the nucleosome, which is this chromatin unit that allows for condensation of DNA. Now, we need to make a step back here. To understand more of the epigenetics, we need to make a step back and go back to these very important proteins called the transcription factors that, like we said, are the fingers that basically activate your switches, where your switches are the enhancers and promoters. There are these non-coding regulatory elements that then turn up, once you turn them on, this results in turning the gene on. And the switches are activated by these transcription factors. So imagine, again, like a finger turning on the light. So what are transcription factors is very important for our conversation today. So transcription factors are proteins with DNA binding capability. So they have some domains. Zinc finger domain is one of them, but there are several domains that are allowed for the binding of the DNA. So these proteins are DNA binding. They can activate or repress gene expression like a finger. They can turn the light on and off, activating the switches. They're very important in eukaryotic cell types, especially during development, when imagine you need to turn on and off genes in a very fine-tuned manner in specific developmental windows. And transcription factors work by having a specific sequence pattern that they can recognize and bind, because there are hundreds, probably thousands of transcription factors in the eukaryotic genomes, and each of them can bind a very specific sequence motif, which is normally 6 to 8 binding site nucleotides that they read and recognize to bind. The problem is transcription factors, the 99% of them, they need the chromatin to be already active and euchromatic state open, nucleosome depleted to be able to bind, like I showed you before. And so how can they activate chromatin if the chromatin is repressed? Who is going to actually dictate the switch from heterochromatin to euchromatin? So this is performed by a specific class of transcription factors. It was discovered by Kenzer, who's now professor at University of Pennsylvania, at UPenn. They discovered this specific family of transcription factors called pioneer factors. Pioneer factors, like the name said, they are the first transcription factors arriving. So your chromatin is still repressed, heterochromatic. But pioneer factors are the only family of transcription factors that are able to bind condensed nucleosomes, repressed nucleosomes. They bind, and this leads to the recruitment of other coactivators, so chromatin remodeling complexes. We will see in a couple of slides what these are. These coactivators will open up the chromatin, and once the chromatin is open, the other transcription factors can arrive and bind. And so the enhancer, your promoter, your enhancer is now activated, And you can now recruit all of the other cofactors that will lead to the 3D looping that we said, and ultimately to activate gene expression. Just to show how it works, your pioneer factor will scan until it finds the motif. It scans the chromatin, it finds the motifs, and by recruiting other cofactor will dictate the depletion of this nucleosome. The DNA will be now naked and free, and the other factors can now bind. And so I said that in order to dictate the elimination of the nucleosome and the eviction of the nucleosome and the opening up of the chromatin, they will recruit these protein complexes. So protein complexes that we call chromatin remodelers because they remodel chromatin composition. And the way they do that is they're big protein complexes made of many subunits, and they all have something in common. They use ATP hydrolysis as a source of energy to do this nucleosome remodeling. So they can slide nucleosomes, evict nucleosomes, exchange histones, add more nucleosomes if they need to repress, so they can have both an activation or repressive function. The most commonly, just one example of that, the most common chromatin remodeler in the eukaryotic cells is called BAF, or switch SNF or SWI SNF, they're synonymous. BAF complex, you can see an example here. Sorry. It's a very large protein complex made of approximately 15 subunits, some of which are mutually exclusive and interchangeable. For example, 81A1B, SMARCA24, and many others. Like we said, it controls nucleosome movement, nucleosome remodeling, nucleosome profiles by using ATP hydrolysis as a source of energy. The 15-20 subunits can be assembled in different configurations, which lead to different types of BAF complex, canonical, non-canonical, polybrom associated, and others. But this is just one of the examples. There are 4 or 5 main families of chromatin remodelers in the eukaryotic genomes. And so to summarize what we said so far, we've discussed about chromatin. We've explained that chromatin is really a tool that the genome uses to keep the DNA really condensed and to be able to fit the nucleus of a cell. And we described the first of the epigenetic mechanisms, which is change in nucleosome patterning and change in nucleosome flagging throughout histone modification, so covalent modification of histone tails, which will flag your chromatin and determine whether the chromatin has to be active or repressed or both. Sometimes you have a bivalent state. So this first type of epigenetic modification, epigenetic tool is the histone modification. And now we will transition to a second type of epigenetic modification, which is DNA methylation. So in the previous case, histones were methylated, right? So the protein was methylated. In this case, instead, is the DNA to be methylated. So DNA methylation is here what we're going to discuss. So the DNA is methylated, and it's a reversible methylation, normally at position 5 of a cytosine, so the C nucleotide, basically, by enzymes that are called methyltransferases. These are particularly common in vertebrates and mammals, this type of DNA methylation, whereas, for example, are more rare in some of the other phyla. For example, in insects are quite rare, in Drosophila it's quite rare. But in mammals and humans, it's very common, this methylation of these cytosines. And usually it's the cytosines that tend to be the most methylated are the ones found in these GPC islands, which are regions of the genome enriched for G and C, so cytosines and guanine. And so these CPG islands are normally methylated, and when methylation happens, this means that the DNA is flagged to be silenced and repressed. So DNA methylation is a repressive epigenetic mechanism to repress chromatin by, first of all, preventing, again, the binding of transcription factors, and to basically keep the chromatin also in a state that is repressed and just statically. And so these are basically 2 faces of the same coin, because if your chromatin is repressed, your transcription factor cannot bind. And so, like I said, this almost exclusively occurs at GCP island. It's symmetrical, so it's normally maintained throughout cell division. There are 2 main enzymes that take care of, 2 methyltransferases that take care of methylating DNA in human cells. DNMT3A and B specifically, and this is just not humans, they are conserved across mammals. Only exception is during mitosis, but we will see that during mitosis, where DNMT1 is instead the enzyme performing this operation. But otherwise, DNMT3A and B are the dominant ones normally in somatic normal conditions. So which regions, so where are these GPC islands located and which regions are normally methylated the most? So intergenic regions tend to be methylated. So outside genes, you tend to have a lot of DNA methylation where you have CPG island. This can include, for example, enhancers that you don't want to activate, repetitive elements or transposable elements. These are parasites of the genome that need to be repressed so they can't move. And in this case, they get methylated by DNA methylation. And then right upstream of the genes where you have the promoter regions, here, for example, your promoters, when the gene has to be silenced, your promoter is normally methylated. So a methylated promoter means the gene is silenced. Your transcription factor is not going to be able to activate the switch because there is DNA methylation. When you lose DNA methylation from the promoter becomes active and can be bound by transcription factors and can activate the gene. Okay, so we've seen two main mechanisms of epigenetic, histone modifications and chromatin remodeling in general, and DNA methylation. So again, modifications of the proteins that form the nucleosomes and modification, in this case, methylation of the DNA that forms the nucleosome. So histone methylation and histone modification in general and DNA methylation. A third main family of epigenetic mechanism is represented by long non-coding RNAs. So when you think about RNAs, you normally think the messenger RNA that is produced from the body of a gene, from the exons, from the coding exons. These are coding RNAs that then are translated into proteins. But these are just some of the RNAs that you have in the eukaryotic cells. Then you have families of RNA that are not translated into proteins, so they are non-coding RNAs, but they still have very important regulatory functions. And they can be short or small, let's say small non-coding RNAs, or long non-coding RNAs. And today we'll talk about long non-coding RNAs. By long, we mean more than 200 nucleotides. There are thousands in the eukaryotic cells, thousands in the human genome. And they can be involved in gene regulation, in epigenetic mechanisms in many different ways. And we will shortly see an example of them. But they can really be involved in many things. They can be involved in helping with the looping of enhancers and promoters. They can go and repress mRNAs, or they can perform other functions, recruit proteins at some specific loci, and so on and so forth. And we will see now what's the most widely studied long non-coding RNA and its function. So this specific long non-coding RNA is called EXIST. And it's involved in a very important process, which is inactivation of the X chromosome. So this happens only in, so let's talk about humans, this happens only in females, because remember, males have one copy of the X chromosome and one copy of the Y chromosome, whereas females have two copies of the X chromosomes. And you need to maintain a balance in the expression of the genes that are on the X chromosome. Since humans only, males only have one, females also need to express the genes from one of the two X chromosomes, which means the other one has to be entirely repressed. So the entire one of the two X chromosomes in females needs to be entirely repressed. And this repression is coordinated by this very important long non-coding RNA, EXIST. So this is a very long RNA, 17 kb long. And right after mitosis, right after cell division, this long non-coding RNA will be expressed by only one of the two X chromosomes. And which chromosome will express EXIST will determine which chromosome will be inactivated, because EXIST works in cis, which means works on the same chromosome where it's transcribed from. And so the X chromosome that will transcribe EXIST will then inactivate that, will be inactivated by EXIST itself. So how does that work? So you can see, first of all, here the high expression of EXIST. EXIST has a domain which allows for the interaction with the PRC2 complex that we have introduced before, right? This polycomb repressive complex, which again deposits this repressive histomethylation. So what EXIST does is will interact with PRC2, bring it to the X chromosome, and this will lead to deposition of this repressive methylation, histomethylation, to the entire chromosome. So the entire chromosome will be histomethylated and completely inactivated and silenced. And this is again determined by this London coding RNA, which is transcribed from the chromosome itself. It acts in cis, so on the same chromosome, by recruiting this protein complex. And this will result in deposition of the methylation. Now, we've seen like three main epigenetic mechanisms that are histomodifications, DNA methylation, and then London coding RNA. We've seen the example of EXIST, but there are thousands of London coding RNA with similar functions. But these are just the main epigenetic mechanisms, but there are some other epigenetic tools that need to be discussed today because they're very important for the activation of gene expression. So I told you at the beginning that enhancers and promoters need to interact with each other by 3D looping. They need to be in close proximity to activate a gene. But so in the human organism, in the human genome, we have probably close to a million enhancers, and something around 100,000 promoters. So can enhancers and all the enhancers and all the promoters interact with each other, or are there some constraints? The answer is there are some constraints, some very specific tridimensional constraints in the chromatin that prevent some enhancers from interacting with some promoters. And these constraints, these 3D constraints, are determined by specific domains that are called topologically associated domains, or TADs. These are fundamental units of the chromatin, tridimensional unit of the chromatin. You can imagine a TAD like being an apartment, a flat of a big apartment complex, right? So imagine like your DNA, your entire genome is a big apartment complex with many apartments, with many units, and each TAD is an apartment, each TAD is a unit. And then only people that are in the same apartment can talk to each other, whereas people that are in different apartments cannot. And so just replace the people with enhancers and promoters. So enhancers and promoters are in the same apartment, in the same TAD, they can interact. Enhancers and promoters that are in different apartments, in different TADs, they cannot interact. And this is very important. And I'll show you in just 2 slides how this is regulated. So the blue here is just your genome. And to form a TAD, you need 2 main proteins. One is called cohesin, and one is called CTCF. So cohesin is a ring, has the shape of a ring. What cohesin will do, it will form a loop here, and it will start pulling your DNA outside of the ring. And this is the TAD that is being formed. And cohesin will pull and pull and pull until, and you see they are in green, 2 CTCF molecules. CTCF is the transcription factor, which will recognize a specific binding motif here. CTCF will bind here and here. Cohesin will pull and pull and pull until you can see the 2 CTCF molecules will meet each other. And this is called TAD boundary. TAD boundary is determined by binding of CTCF and cohesin. And so all of the enhancers and promoters that are found in here will be able to interact with each other. But if you have an enhancer here and a promoter here, the interaction will not be possible. So the promoter of the gene here will not be activated by this enhancer. So this enhancer will not be able to activate a gene that is found here, and vice versa. This model is called loop extrusion model. And again, is underpinned by cohesin, this ring here, extruding chromatin until 2 molecules of CTCF meet and close it, the TAD boundary. So this is just an example of, this is your TAD, this is another TAD. You have this enhancer cluster here, a gene here with its promoter, another one here with its promoter. So the enhancer will be able to activate the red gene, but not the PAX3 gene here, the blue one, in normal condition. But let's say you have a mutation at the TAD boundary, which breaks the TADs, TCF can no longer bind. And so your enhancer here will aberrantly activate PAX3. And if this happens during development at the runtime point, you can see how this can result in quite severe consequences. Just a TAD boundary broken. Okay. So again, this is not really a modification of the chromatin itself, but is and certainly is not a modification of DNA. But the TAD the TAD formation, this 3D unit formation, is another way of the genome to regulate gene expression by altering the function of the gene. It will determine which gene is expressed or not by isolating the gene from some enhancer or by putting an enhancer and a gene in the same domain, allowing for interaction and gene expression. And the last tool I wanted to show you today before going the last few minutes talking about methodologies, the last important component of gene expression is certainly RNA polymerase II, which is this super highly conserved, very large enzyme, which literally transcribes mRNA from exons, right? So you have your DNA sequence, and your polymerase II will transcribe the DNA into mRNA, and then the mRNA will ultimately go into the ribosome and be translated. And what we need to know about POL2 is just there is not a like a continuous the transcription of POL2 is not a continuous process, but it has some there are some important factors and modifications again, which can be also they can also fall under the umbrella of epigenetics, that are also very important in determining how a gene is expressed or repressed, and with which timing as well. And what we're going to focus today specifically is the posing of RNA POL2, which we'll see now in a couple of slides what it is. So RNA POL2 is a very large enzyme, which is in a very core domain here. And then it has the C-terminal domain, or CTD, which is made of repeats, which in humans are these 7 amino acids here, for example. And you have dozens of these repeats, approximately 52, for example, in the human genome. So these are humans, this is yeast. You can see they're mostly conserved. So you have these repeats that are very important, and they tend to be highly conserved. And why are these repeats important? Because phosphorylations of these different repeats will work as flags. So it's something very similar to the histone modification. Here is not the histone to be flagged, is the POL2 protein that is flagged, and specifically the C-terminal domain. And different types of phosphorylation will determine if the POL2 has to stay steady and not move, or if instead the POL2 can move. These are just some examples: serine 5 phosphorylation, serine 7 phosphorylation, tyrosine 1 phosphorylation, and so on and so forth. You can see that this is your transcription start site, and this is the end site, so beginning of the gene, end of the gene, and your POL2, initiating POL2, elongating POL2, and terminating POL2. They are flagged with different phosphorylations and modifications that will determine the state, the transcriptional state of the POL2. And what's really important in this process is understanding what specifically is RNA POL2 promoter proximal posing, which is a mechanism that keeps the POL2 stuck at a specific site during transcription. It was discovered in 1986 in Drosophila, but it's widespread everywhere in eukaryotes, basically. And it was discovered in early response genes, but now we know that it's widespread genome-wide as well. And it's crucial to be able to transition quickly and productively from initiation to elongation. And I'll show you in a moment how this works. So your RNA POL2 arrives at the promoter at the transition start site and start producing mRNA, messenger RNA. But then this transcription is arrested normally after about 50 base pairs, where RNA POL2 enters in a so-called post configuration. And the post configuration is decorated, is flagged by serine 5 phosphorylation in the CTD. So the serine number 5 gets phosphorylated, and your POL2 gets stacked, and it is kept stacked, and it can't move. And there are two main proteins that contribute to keep the POL2 stacked in there. One is called NELF, and one is called DCIF. So these two protein complexes specifically keep your POL2 there. Your CTD is phosphorylated as serine 5, and this prevents POL2 moving. So why is this important? Because your POL2 is posed, but this also means that it's poised. So what it means is that you're doing development. This gene will need to be expressed quickly at some point. And so rather than having to load POL2, and for loading POL2, you need a lot of proteins here, and transcription factors, load pol 2, start transcription, and complete transcription all together, this is a very time expensive and energy expensive process. Instead, you start transcription no matter what, and then you keep your pol 2 here at a leash, ready to go. It's like you have a gate, and then when your gene needs to be, finally transcribed and expressed, you quickly dephosphorylate serine 5, phosphorylate a different residue serine 2, remove the alpha, phosphorylate the SIF, which will travel your pol 2, and then it's like shooting with a gun. Your pol 2 is now ready to go and will very quickly produce mRNA and express the gene. And this is very useful, particularly during development. It's very useful when you have early response genes that need to be transcribed super quickly. So instead of having to load the entire machinery, load pol 2, start transcription, pol 2 is already there. It's been already pre-processed. It's just good to go. You just need to remove the alpha and phosphorylate the SIF and phosphorylate the CTD. Your pol 2 will just, jump the gun and go. And this is a very, very, very important mechanism to express genes quickly and timely. And so just to conclude the first part, and then just a few minutes will be dedicated to the second part. So we have studied epigenetics. We have understood what epigenetic is. So modifications of the genome, which will not alter the sequence of DNA, but will alter the chemical, physical, biophysical structure of DNA to, trigger specific patterns of gene expression, gene silencing, and so on and so forth. And we've seen a few examples. We've seen histone modifications, DNA methylations, long non-coding RNA, TAD formation, and then RNA pol 2 phosphorylation. These are the main, the main epigenetic actors. But how do we study now in the laboratory? How do we know all of those things? So we didn't know much until 2005, right? We knew, some specific examples of single loci, single genes. We knew, we characterized some of them really well, but we didn't know anything, pretty much anything on, most of the genes and their regulation. But then in 2005, there was the big biotech revolution in genome sequencing, which was what we still refer to as next generation sequencing, which is an approach, a technique that allows to sequence, your entire genome or, specific characteristics of the genome in a few minutes for a very low price. So just to give you an idea, when the first genome, the human genome, was sequenced in 2001, it took many, many years, and it cost millions of dollars to do it, to sequence just one gene. Now, with next generation sequencing, and you can see here some technicalities, but I'm not going to to waste time on this. But basically, with next generation sequencing, now you can study an entire genome. We can sequence an entire genome in an hour or less with maybe $10, $20. So it was a huge revolution, which allowed the scientists like us, the genomicists, to be able to now study the entire genome and its modifications genome-wide, and to make comparative studies, and to do that across species, across individual, and at the population level. It was quite the revolution. So today we will, I will just discuss in the last 5 minutes just a few next generation sequencing derived techniques that allow to study epigenetic modifications. And so we've discussed a lot about histone modifications and transcription factor binding. And so what are the techniques that we now use to study and to identify which histones have a specific modification in the genome, or which transcription factors are binding which hindrances and which promoters? So there are now 2 main techniques to do that. One is called ChIP-seq, or chromatin immunoprecipitation followed by sequencing. And the other one is called cut and run, or there is now a new derivative called cut and tag. They're very similar to each other. And I will discuss them now in a moment. Let's start by ChIP-seq. So if a DNA binding protein like a transcription factor is able to bind DNA, you can use an antibody that recognizes that protein to isolate all of the DNA regions that are bound by the protein itself. And I'll show you now how it works. And just something very important, you can use the same approach. Instead of the antibody for DNA binding protein, you can use a specific antibody designed for histone modifications to identify what the histone modifications are. So how do you do it? So here you have your transcription factor that is interacting with DNA. So this is your so-called chromatin, right? You're going to have to take a snapshot of that. So you're going to have to make this interaction permanent, because normally, obviously, it's very dynamic. But for you to be able to identify this specific genomic site that are binding the transcription factor, you're going to need to take a picture of this. And you take a picture by using a compound called formaldehyde, which will cross-link, that's the terminology we use, cross-link your transcription factor to the chromatin, and will permanently stack them, they will be permanently bound here. And now you have a artifact, I guess, which is a permanent interaction of transcription factor with DNA. Then you can just shear the chromatin and make it into small pieces, which will help antibody binding. Then you can use your antibody, you can see here, to recognize the transcription factor. And so your antibody will pull down all these genomic regions with the transcription factor, whereas it will not pull down genomic regions that are not bound by the transcription factor. So then you can isolate just the DNA by detaching it with specific tools. You can detach the DNA from the transcription factor at this point, because you have discarded already all the genomic sites that are not bound by the transcription factor. You only retain the ones that are with the transcription factor. And then you can sequence all of them with next-generation sequencing. You will obtain short sequence, short DNA sequences that we call reads. Normally, a read is 50, 100, 150 nucleotides of DNA. And you sequence millions of them with that machine that I showed you before, the next-generation sequencer. You will sequence millions of these, and then you will map them to the human genome. And whenever you have a enrichment of reads and map, like a pilot like this, is a peak of reads. Whenever you have a peak of reads, it's because you're picking up these regions that are bound by transcription factors. And then you can just visualize them bioinformatically, and you can see, like, here you have no peaks, which means your transcription factor was not binding. But here you have your pileup of reads, where clearly your transcription factor was binding. And then you can do it for histone modification by using antibodies for histone modification, or transcription factors by using antibodies for transcription factors. It's a very powerful tool, but it has some drawbacks. One is that it requires a lot of cells. For a ChIP-seq experiment, you need 10 to 20 million cells, which is a lot. And so sometimes it's very hard if you're working with some tricky cell types. Let's say you're making neurons from stem cells, for example. It's very hard to get that many cells. Also, cross-linking and chromatin sharing, which we call sonication, can introduce a lot of variability. And then obviously it relies on antibody quality, because if your antibody is not good to pull down your transcription factor of interest, then it won't also pull down the DNA that is bound to it. And so your ChIP-seq will not work. And so the other technique, cut and run, that's been developed a few years ago, it will fix some of the issues, so specifically the first two issues. Unfortunately, it still relies on antibodies, so you can't really solve this problem. But the first two problems have been solved by the introduction of cut and run. So cut and run, instead of using cross-linking of the DNA and transcription factors together, it just works in native normal conditions, and it uses a protein called MNase, which is able to diffuse in the cells. And MNase is like a scissor. What MNase does is cuts and chews DNA. And what we use is an MNase, which is fused with the antibody, with a protein A, which is a protein that can bind with the antibody. So you pre-treat your MNase protein A, it's called protein A, this specific protein. You will pre-treat it so that it will interact with the antibody. And now you have your protein A MNase here, binding with the antibody. They will diffuse into the cell and will bind wherever the antibody recognizes the transcription factor. And then the MNase here, acting like scissors, will cut left and right here, you can see it, and detach exactly that piece of DNA that is bound by the transcription factor. And this will then be collected and sequenced with the same approach, next generation sequencing, to then get the same thing, peaks, right? So the visualization and the analysis is going to be the same. So what's the advantage of cut and run over ChIP-seq? One is that you don't need to do any cross-linking and sonication, which introduces technical variability. And most importantly, you don't need 10 million cells. You can do it with 100,000 cells. And actually, now there are kits that allow to do cut and run also a single cell basically level, or with just a few thousand cells. So this was obviously a big revolution in the field. And again, to summarize, ChIP-seq and cut and run and cut and tag, which is very similar to cut and run, just uses a different enzyme. They allow to profile binding of transcription factors and to profile histone modification. So very important epigenetic features. Then the next two slides, they're going to be devoted about on methods that allow to detect regions of open chromatin, so euchromatic regions. So how do I know which regions of the genome are nucleosome-free, open, active? So what is euchromatin and what is heterochromatin? How do I know? There are a few techniques that have been developed, and today we will discuss mostly attack-seq, which is the most commonly used, the most efficient, and the ones that everyone uses these days. So again, you have your heterochromatin, and then your euchromatin here. Euchromatin again is you remove the histones, your DNA is free and naked to be bound by transcription factors. And so this is used in our advantage because there is this enzyme, which is called TM5, which is an enzyme that is only able to interact with naked euchromatic DNA. And the enzyme, similar to the MNase, it has exonuclease property, so it will cut DNA, but will only cut DNA when the DNA is naked. So when DNA is euchromatic, it will cut it, and when it cuts, it also inserts some adapters on the sequence, and the adapters will be used to then make this next generation sequencing libraries to sequence them. So you're going to have all these fragments coming out of your experiment, and all of the fragments that are coming out of the experiments are all euchromatic active fragments, whereas this will be untouched. So when you sequence, we're only going to sequence this. And then the results is very similar. You sequence, you're going to have your reads, and then you map them on the genome, and your reads will pile up only in case of euchromatic chromatin, because your TM5 will only be able to cut euchromatic chromatin. And then you visualize very similar to ChIP-seq. You can see peaks where the chromatin was accessible. And then you can look at the sequence of these peaks, identify motif of transcription factors that are enriched in this euchromatic region, so you can predict which transcription factors will bind. And then you can look genome-wide with heat maps, and you can do a lot of different analysis. A similar tool you can use to determine which regions of the genome are active is the MNase-seq. So it's similar to the attack-seq, but instead of using TM5, it uses MNase, which was the same enzyme we just discussed for the cut and run. MNase will just similarly just cut wherever the DNA is free, and it will not just cut, it will chew it, it will digest it, it will get rid of it. So MNase will get rid of all of the accessible naked DNA. So what you're left with is just nucleosomes in this case. And then you sequence it, and basically, instead of identifying peaks of accessible chromatins, your MNase peaks will be locations of nucleosomes. So you specifically use it to determine nucleosome positioning rather than to determine where chromatin is accessible. So it determines nucleosome positioning, whereas the attack-seq determines chromatin accessibility. So similar but slightly different techniques. And then, and this is the last one, I believe, how do we identify regions of the genome where DNA is methylated? Remember, we said DNA methylation is one of the main epigenetic mechanisms, and it's used to repress regions of the genome, so to repress promoters, to repress transposable elements, for example. And like we said, it's methylation of a specific cytosine, cytosine 5 in GPC island especially. So the DNA methylation is started using bisulfite sequencing. And so how does this work? So bisulfite is a compound which will convert the cytosine, so the Cs, into uracil use. So remember that in the DNA, normally you don't have uracil. Uracil is typical of RNA, right? Whereas in DNA, you have Cs. And so what will happen is that your bisulfite will convert cytosines into uracil, but when you have DNA methylation, your cytosine will not be affected by this and will remain a cytosine. So whenever your C is methylated, remains a C. When your C is not methylated, your bisulfite will transform into a U. Again, U's are not present normally in DNA, but just in the RNA. So you treat with bisulfite, you sequence the whole thing, and then you map the reads onto the human genome. But since you're going to have a lot of U's, your reads will not be able to map because there is no U's in the human genome, so they will be discarded. So only the reads that are able to map will be the ones that have retained the Cs, which means they're the ones that were methylated. So the only reads that we're able to map are the methylated reads. So the reads that come from methylated DNA. And then again, you're going to have bilateral reads, peaks, in regions of the DNA that are methylated. And this is the only, the most reliable tool to identify DNA methylation. So once again, you convert the C into U's with bisulfite, but the cytosines that are methylated will not be able to be converted. And so these are the reads coming from here, they are going to be the only ones that are going to be mapped to the genome. And so this allows you to profile where DNA methylation is. So with this, I'm done. I just want to summarize what we have discussed today. So we've learned what epigenetic is, and we learned that it's instrumental to regulate gene expression without affecting DNA sequence. So these are chemical, physical, biophysical, structural modification of the DNA, the genome, that don't change the sequence of DNA, but they're instrumental to determine which gene is expressed, which gene is not, and to what extent you need to express a gene. We've discussed some of the main epigenetic mechanisms, such as histone modification, DNA methylation, and non-coding RNAs. And then we've also briefly discussed, these 3D units of the chromatin, the TADs. And we've discussed how RNA-pore2 can also be modified to affect gene expression, for example, throughout phosphorylation of the C-terminal domain. We've discussed transcription factors and what they are, and the specific example of pioneer factors that are the first ones scanning and arriving to the chromatin, recruiting the chromatin remodelers, which we have also discussed, to establish chromatin states. And finally, we've discussed in the last few minutes next generation sequencing and some important techniques that can be used to study epigenetics. And with this, thank you for inviting me to teach this lecture, and I will stop this share.